Hello everyone, this is Dr. Manasvi Pandey and I'll be taking your lecture on the topic gastrointestinal tract and liver. So let's get started. The gastrointestinal tract is the portal through which the nutritive substances, vitamins and minerals and fluids enter the body. It is more than 10 meters or 30 feet long, starting from our mouth, passing to the pharynx, the esophagus, the stomach, the small and large intestine, and ending in the rectum and anus. Associated with it are the accessory organs of digestion which help in digestion, such as teeth, tongue, salivary glands, liver, and pancreas. So let's look at the pictorial representation of our digestive system, this diagram. So the digestion process, like as we think digestion, the common concept is that digestion happens inside the stomach. No, the digestion starts as soon as we consume the food, as soon as the food enters our mouth. So mouth helps in chewing the food and it mixes the food with saliva. Salivary glands produce saliva which contains salivary amylase which help in the digestion of starch. Pharynx, it swallows the chewed food mixed with the saliva, which is called bolus. Esophagus moves the bolus to the stomach, so it is the passage through which the bolus moves and reaches our stomach. The stomach mixes and churns the food with hydrochloric acid and pepsin, which is a protein digesting enzyme, creating chyme. The liver makes bile juice, secretes bile juice, which aids in the digestion and absorption of fat or emulsification of fat. The pancreas releases bicarbonate to neutralize intestinal contents, produces enzymes that digest carbohydrates, protein, and fats. The gallbladder here stores the bile juice secreted by our liver and it releases into the small intestine or the duodenum as and when it is required. And the sphincter of Oddi here controls when it has to open or not, and accordingly the bile juice is released into the small intestine. And the small intestine digests the food and absorbs nutrient into blood or lymph. Large intestine absorbs the water and some vitamins and minerals, and it is also the home to intestinal bacteria, and it helps in passing away the waste material outside our body. Anus opens to allow waste to leave the body or in the process of defecation. What are the functions of digestive system? So the GIT is responsible for breaking down food and supplying the body with water, nutrients, and various electrolytes needed to sustain life. This is brought about by a few processes. The first is ingestion, then digestion, absorption, egestion. So let's look in detail. The ingestion process involves placing of the food into the mouth, chewing the food into smaller pieces with the help of our teeth or the mastication process, moistening the food with salivary secretions, swallowing the food or deglutition. The digestion process is the process when the food is broken down into smaller particles by the grinding action of the GIT and then degraded by our digestive enzymes into usable or utilizable nutrients which our body can actually use. Absorption. During absorption, the nutrients, water, electrolytes are transported from our GIT, mainly from the small intestine, to our normal circulation. Egestion process is the removal of waste from our body. So during egestion, the undigested food along with various secretions and sloughed off epithelial cells from the gastrointestinal tract pass into the rectum and constitute the feces which are voided through the anus at periodic intervals. Now the digestive organs are of two types: primary digestive organs and the accessory digestive organs. Primary digestive organs are the organs where the actual digestion takes place, and the accessory digestive organs are those which help the primary digestive organs in the process of digestion. The primary digestive organs are mouth, pharynx, esophagus, stomach, small intestine, and large intestine. And the accessory digestive organs are teeth, tongue, salivary glands, exocrine part of pancreas, and our liver. Now, what is the organization of the structure of the GIT? So, various walls the layers of GIT. So here we can see in this diagram the outermost layer, like starting from the outermost layer, we have serosa. Then we have the layer called muscularis externa, which comprises of longitudinal smooth muscles and circular smooth muscles. Then the submucosal layer is there and the mucosa layer is there, which comprise of muscularis mucosa, lamina propria, and the lining epithelium, which has these finger-like projections known as villi, which help in propulsion of the forward movement of the food. And inside here, this is the lumen portion. So let's look in detail about these layers. The serous layer, in general, the organization of the wall of GIT from posterior pharynx to the anus has the following layers from outside to inwards. The serous layer or the serosa is useful in attachment of the gut to the surrounding structures. Then we have the muscularis externa, which comprises of longitudinal and circular smooth muscles. The circular smooth muscle layer is thicker at the sphincter. The longitudinal and circular, these both smooth muscle layers are useful in local mixing and forward propulsion of the contents of the gut. The submucous layer consists of loose connective tissue, blood vessels, and lymphatics. The mucous layer is lined by epithelium and consists of a stroma containing various glands, muscularis mucosa of smooth muscle, loose connective tissue rich in lymphocytes. Innervation of GIT, that is the enteric nervous system or the little brain. Now this enteric nervous system, why is it called the little brain? Because it contains more than 100 million neurons and most of them are found in the spinal cord. So there are two major network of fibers which innervate the GIT. They are intrinsic and extrinsic. Together, this intrinsic and extrinsic nerve fibers constitute the enteric nervous system or the little brain. Intrinsic innervation comprise of myenteric plexus and submucous plexus or Orbach plexus or Meissner plexus, and the extrinsic innervation comprises of parasympathetic and sympathetic or cholinergic and adrenergic innervations. The intrinsic innervation has myenteric plexus or Orbach plexus, which lies between the longitudinal and the circular smooth muscle layers and innervates both the layers. It is mainly motor in function and the submucous plexus, whereas it is sensory in function. So the myenteric plexus controls the motor function and submucous plexus does the sensory function. It's the myenteric plexus stimulation increases the activity of gut by increasing the tone of gut wall, increasing the rate and intensity of rhythmic contraction, and thus it is concerned with the control of peristaltic activity of the gastrointestinal tract. The submucous plexus or the Meissner plexus lies between the submucous layer and the inner circular smooth muscle layer. It has sensory function, as I told earlier, and it is concerned with the control of exocrine and endocrine secretions by the cells in the gastrointestinal tract. Now, this was about the parasympathetic, and now let's look. No, okay, yeah, here. We just got done with the intrinsic innovation. Now let's look into extrinsic innovation. The extrinsic innovation is under the autonomic nervous control, that is parasympathetic and sympathetic. Parasympathetic or the cholinergic nerves release acetylcholine at their endings by the process known as depolarization of smooth muscle membrane, which produces the contraction of the GIT muscles. Stimulation of parasympathetic nerves to the GIT produces increase in motility and tone, relaxation of our sphincter, increased secretion from the stomach, especially of various enzymes and of the intestine. The sympathetic or the adrenergic nerves release epinephrine at their endings by the help of hyperpolarization of smooth muscle membrane, which results in relaxation of GIT musculature. The stimulation of this sympathetic or the adrenergic nerve to GIT produces decrease in motility and tone, contraction of sphincters, and inhibition of secretions from the stomach, and probably inhibit the intestinal secretions as well. Now let's look deeper about liver, like what are the functions, what is the structure, the parts of liver. So here the location. So here we have our diaphragm and this is our stomach. The liver is located just beneath the diaphragm and just above the stomach in the upper right side of our abdominal cavity. So this is about the location of liver. Liver is the largest gland in our human body, which weighs around 1500 grams, which is quite a lot because this is 2.5% of our total body weight. And the color is reddish brown and shape here you can see it is wedge shape. Now the hepatic lobes, various parts. So the hepatic lobes are four in number. If we look anteriorly, it is having right lobe and left lobe. Right lobe is the larger one and the left lobe is the smaller lobe. And if we see through the posterior view, we can see two lobes again, quadrate lobe and the caudate lobe. So the quadrate lobe is here and the caudate lobe is here. So in total, we have the liver has four lobes. Now we are looking at hepatic lobules. Previously it was hepatic lobes. Now we are going to see about hepatic lobules, which are the structural and functional unit of liver, and they are about 50,000 to 1 lakh lobule in number in our liver. This lobule is a honeycomb like structure and it is made up of hepatocytes or the liver cells. The hepatocytes or the liver cells and the hepatic plates. So the hepatocytes are arranged in various columns which form the hepatic plates. Each plate is made up of two columns of cells and in between the two columns of each plate lies the bile canaliculus. So here we can see all this in the picture. In between the neighboring plates, a blood space called the sinusoid is present here. The sinusoid is present and sinusoid is lined by endothelial cells. In between the endothelial cells, there are the special macrophages known as Kupffer cells. It is very important. The Kupffer cells are present between the endothelial cells, and the function of these Kupffer cells is that they destroy the microorganism in the blood. Here also the Kupffer cells are there in this picture and here also. So these. Okay. Now the portal triads. Each lobule is surrounded by many portal triads, and each portal triad, like triad, comprise of three vessels. One is the branch of hepatic artery, a branch of portal vein, and a tributary of bile duct. So, yeah, here this is the hepatic artery branch, this is the portal vein, and this is the bile duct. Blood supply to liver. Now, our liver is supplied by hepatic artery, portal vein, and hepatic vein. Hepatic artery arises directly from aorta and supplies the oxygenated blood to liver. So see this in this picture, this is a very good representation, very good flow chart to remember this. Hepatic artery carries the oxygenated blood and the portal vein carries the deoxygenated blood. This is what is there in the liver and this is what is there after leaving the liver. So the hepatic artery carries the oxygenated blood, portal vein carries the deoxygenated blood, and these two together come like meet in the sinusoid. So it like let's look in detail. It arises directly from aorta and supplies oxygenated blood to the liver, and the portal vein brings the deoxygenated blood from stomach, intestines, spleen, and pancreas. The blood from hepatic artery mixes with the blood from the portal vein with the deoxygenated blood. So here the oxygenated and deoxygenated blood mix together in the hepatic sinusoid. The hepatic vein. The central veins from many lobules unite to form bigger veins, which ultimately form hepatic veins, which is right and left, which opens into the inferior vena cava. So the hepatic vein opens into the inferior vena cava and goes to the right atrium. Okay. Now, what are the functions of liver? Metabolism, breakdown of erythrocytes, and defense against microbes, Kupffer cells we have seen previously. Detoxification of drugs and toxic substances. inactivation of various hormone, production of heat, secretion of bile, juice, storage of substances. Let's look in detail. It helps in metabolism of carbohydrate, fat, and proteins. The glucose is converted to glycogen, which is the form of storage, and the glucagon stimulates the conversion of this glycogen into glucose. So, thus, it helps in maintaining the blood glucose levels. Fat metabolism. Okay, liver helps in fat metabolism also because the stored fat can be converted to energy. Protein metabolism, deamination of amino acids, and transamination, synthesis of plasma proteins. These three processes, these are all carried out in the liver. Breakdown of erythrocytes and defense against microbes by the help of Kupffer cells. Detoxification of drugs and toxic substances. Now, these include the alcohol or the ethanol, waste products, and microbial toxins. This is because after the absorption from the alimentary tract, they travel in the blood to the liver where they are largely metabolized so that the levels in the blood leaving the liver and which enters the systemic circulation are inadequate to achieve the therapeutic effect. This is also known as first pass metabolism. So, this is a very interesting concept. Like first the thing the like suppose we have taken 500 mg of paracetamol, so it's not the whole 500 mg of paracetamol that acts in our body that is left to act in our body, because first this drug goes to our liver, and then by the help of first pass metabolism by the liver, a huge fraction of it just gets out, and only a very little fraction of that 500 mg is actually utilized by the body. So this is the first pass metabolism. Function of liver. Okay, inactivation of hormone. It helps in inactivation of glucagon, cortisol, aldosterone, thyroid hormones, sex hormones, and insulin. It helps in production of heat. It helps in secretion of bile. The hepatocytes synthesize the constituents of bile from the mixed arterial and venous blood in the sinusoid. This includes bile salts, bile pigments, and cholesterol. We'll read about it later. Storage of substances include what are all are stored. So these are glycogen, fat soluble vitamins like vitamin A, D, E, and K, iron, copper, water soluble vitamins like vitamin B12. Now, bile juice. We'll look in detail. Bile juice is secreted by the hepatocytes or the hepatic cells of the liver, and it is stored here in the gallbladder, and then it is secreted into the duodenum of the small intestine through the hepatopancreatic ampulla through the bile duct. So first it is secreted here in the liver, then it is stored in our gallbladder, then through this pathway, that is the hepatopancreatic ampulla and the bile duct, then it goes to the finally goes to the small intestine or the duodenum. The volume of the amount of bile juice that is secreted daily is around 800 to 1200 milliliter per day. Now, composition of bile. Bile is composed of 97.6% of water and 2.4% of solids. These solids are further divided into organic and inorganic substances. Organic substances are bile salts, bile pigments, like bilirubin, biliverdin, cholesterol, fatty acids, lecithin, mucin, and inorganic substances are sodium, calcium, potassium, chloride, bicarbonates. So we'll see what are the bile salts. In the small intestine, they help in emulsification or breakdown of fat, which helps in digestion. And the fatty acids are normally insoluble in water, which makes it very difficult for our body to absorb through the intestinal wall. So, what does the bile salt do? It makes the cholesterol and the fatty acids more water soluble, which enables both of them and the fat soluble vitamins like A, D, E, and K to be readily absorbed by our intestinal wall and be utilized for our purposes. Now, what are the bile pigments? Bile pigments are the excretory products in the bile, which are mainly two: bilirubin and biliverdin. Bilirubin is the yellow colored pigment and biliverdin is green color pigment, and these are the two bile pigments which are formed during the breakdown of hemoglobin, and it is having antioxidant properties. So that's all about the gastrointestinal tract and liver. Thank you so much. I hope you understood the lecture.